Okay, cool. Thanks for the introduction, Evan. Hi, everyone. My name is Michelle, and today I'll be presenting some of the work that I've been doing in Emma Farley's lab. So for my thesis, I'm interested in studying how you transform from a single cell all the way to a multicellular organism. In particular, I want to study how this developmental process occurs and how it's regulated. For the project I'll be presenting on today, I was interested in studying how transcriptional changes impact an organism's development. And we can start to answer these sorts of questions with something called cell lineage studies. So cell lineages are the pattern of cell divisions in the development of an organism. It's the process that underlies how a single cell zygote can transform into different tissues. Cell lineage analysis is the tracing of these cellular genealogies and following cell divisions over time as they form different populations. This is an important field of study in developmental biology for several reasons. One, understanding how cells divide and form different tissues will allow us to address the developmental basis of specific phenotypes, such as those caused by mutations, and figure out at what point in a cell lineage did a certain perturbation take effect. These analyses can also help us understand the pattern of cell divisions, such as the polarity of cells in the formation of embryos, as well as help us understand how developmental processes differ or are similar across multiple organisms, especially in organisms that are evolutionarily related to one another. Finally, we can use cell lineage studies to understand the cellular expression patterns of certain marker genes that are important for the formation of specific tissue types. For this project I'll be talking about today, I'll be using a method called single-cell RNA sequencing to understand how specific genes define different tissue types during early development. So single-cell RNA sequencing is a variation of traditional bulk RNA sequencing, and bulk RNA sequencing is a genomic approach that's been used to detect and quantify mRNA transcripts in a biological sample. It's been used to understand cellular responses across many different cell types and how certain genes are over or underexpressed across different populations. This technique is usually conducted on samples containing thousands to millions of cells. However, this method doesn't allow us to evaluate cells directly, and instead compiles a bulk analysis of a tissue. On the other hand, single-cell RNA sequencing is a newer technique that allows you to evaluate the transcriptomes of individual cells within a single tissue, or even a whole embryo. This new ability has enabled researchers to evaluate the similarities and differences of genes expressed within a population of cells, and it's even helped people discover new and rare cell subtypes that we've missed with bulk analyses. Single-cell methods can be used as a way to understand how certain marker genes are important for the formation of specific tissue types, and it provides a modern way to perform cell lineage studies of developing embryos. So I'll be using this technique to study the early development of an organism called Ciona intestinalis. So what is Ciona? Ciona is a part of the urochordates, the sister group to vertebrates, and vertebrates include many organisms that we're familiar with, such as mice, dogs, and us humans. While Ciona may not look like us at all in its adult form, the embryonic form actually shares many of the same tissue types, such as muscle, endoderm, epidermis, neural, and notochord tissue. Additionally, the developmental programs of many of these tissues is conserved with vertebrates as well. Another benefit of Ciona for cell lineage studies is that their cells rapidly develop in a stereotyped manner, so that for every cell, we can tell where it came from and what it will become. The entire course of development for Ciona from the single cell stage to the hatching larva stage only takes around 18 hours. Additionally, organisms at this final hatching larva stage only have around 2,500 cells. The simple body plan of Ciona makes it easy to map all the cells of an embryo, and their fast development make them a feasible organism to use for cell lineage studies. These advantages together make Ciona an ideal model to map the transcriptomes of all the cells in a developing embryo to understand all of the genes that are expressed across development. So now I'll be going through how we use single-cell RNA sequencing to study two important stages in development that are shared between Ciona and vertebrates: gastrulation and neurulation. Gastrulation is a major step in development for all multicellular organisms. At this stage, the embryo begins to implement the body plan that defines what should be kept on the inside of your body and what should be kept on the outside. In Ciona, this is the migration of the endodermal and mesodermal cells inside of the embryo and defining of the outer epidermal layer. Neurulation occurs immediately after gastrulation, and it involves folding of the neural plate into the neural tube to encourage development of the brain. So I collaborated with another grad student in the lab, Ben Song, to run these experiments to look at these three different time points. To sequence these time points of interest, Ben starts off by fertilizing Ciona embryos and then waiting until they're developed to the stage that we're interested in. After that, he can disassociate the embryos at the stage and then run them through the single-cell sequencing protocol. I can then jump into the process and take the sequencing data to analyze to evaluate what genes are expressed across each and every cell in the embryos that we harvested. After determining what genes are expressed across each cell, I can then computationally generate a map with the cell types that are present. So in these maps that we were able to generate, each of these clusters contain cells that have transcriptional profiles that are similar to each other, so that each of these clusters represents the cell types that are present within the embryo. While we'll be performing this experiment on these 3 different time points here, today I'll just present our single cell map that we generated from this stage right here, the late gastrulate embryo. So before I show you the result of annotating our single cell map, I wanted to show a diagram of the embryo at this time point, along with the cell types that we would expect to see in different colors. The single cell maps that we're able to computationally generate are in 2D, but these embryos are in 3D, so it's important to consider where things are located next to each other and what we would actually expect to see. So within the single cell map, we can see the muscle cells here in yellow, as well as the heart beginning to form in gray, as well as the well-defined neural plate rows that we would expect to see. And these plate rows would are going to develop into like the nerve cord, the brain, and other tissues that will be present later on. To contextualize this embryo a little bit, you can think of the embryo as a raspberry because it's in 3D. We have our hole in the center where we can see the layer of fruit on the inside, but we can also see the layer of fruit on the outside. The embryo at this late gastral stage is very similar. So here's our hole, denoting the hole of the embryo where we can see the layer of cells on the inside, as well as the layer of cells that are on the outside of the embryo at this stage. Now that we've contextualized the embryo a little bit in 3D, I want to show the single cell map that we were able to construct from our sequencing data. So here from the single cell map, we have the different cell type clusters that are present. So we have the notochord tissue here, epidermis, as well as muscle, and different lineages of the mesenchyme and endoderm. But we're also able to evaluate different cell type populations that are smaller, such as the different rows of the neural plate, as well as the heart tissue that's present at this stage. To sanity check this data that we're able to determine, we can also compare the ratios of cells that we found in this computational map of the embryo. For example, the number of cells in the epidermis versus the number of cells across the entire embryo, to ratios of cells that we would expect to see from previous Ciona cell lineage studies. So in this first column, I have the different clusters that are present, and then I have the number of cells that we have in our single cell atlas, as well as the percentage of the embryo that these clusters take up. I also have the different percentages of the embryo that we would expect to see for these different cell types based off of the literature. From this chart, we can see that we're able to recover some cell populations at ratios we would expect to see, such as certain populations that will develop into the nerve cord, sensory vesicle, and pigment cell precursors within Ciona. Additionally, there are also some populations we've annotated with more cells than we would expect to see, such as with the notochord, some neural populations, and germ cells. We were also able to recover less than we would expect to see in the developing heart. It's possible that for these clusters I've indicated with these red arrows, we didn't annotate things properly in our computational map, or that during disassociation, these cell types were recovered at a higher or lower frequency. As our first time running single cell in the lab, and given that we're able to recover cell types we would expect to see, we're fairly confident in the data, but ideally, we want to be able to use these maps for something more, instead of just looking at what cell types are present. We want to use these maps to identify all of the cell types in the embryo, but also the genes that are present within these cell types, in order to get a fuller picture of what defines a cell's identity during development. So in addition to checking if markers identified within sera are present in our single cell maps, we can also determine if they're present in actual embryos to see if they make sense with what's currently known about Ciona embryogenesis. So for this experiment, I collaborated with an undergrad in the lab, Hannah, to evaluate if certain markers were expressed in the notochord in real embryos. So brachyury is a gene that's expressed exclusively in the notochord precursor cells of Ciona, and previous work has also determined that brachyury is a critical determinant of the Ciona notochord. Interestingly, brachyury notochord differentiation is actually essential to all vertebrates that's been studied thus far, including frogs, zebrafish, mice, and humans. So with this information in mind, we wanted to use brachyury and other known markers like it to validate our cell clusters. So here I have a figure of our single cell atlas, along with where we would expect the notochord cells to appear on an embryo model. The notochord at this point is on the interior of the embryo. So if we bring back our raspberry analogy, we have a hole around this area that we're peeking in to see where these notochord cells are on the interior surface. When we were annotating our clusters, we first tried to look for brachyury in the notochord cluster, and we see that it's present there. So here we're looking at the expression of brachyury in the notochord cluster, and with this violin plot, we're seeing the degree of expression across the different clusters. And additionally, we wanted to check the expression of other known markers, such as orphan BHLH1, another established notochord marker and nervous system marker. When we evaluate the in situs for these two markers together, we can see that they're where we would expect them to see, so in this notochord grouping of cells. To orient you, here's our whole of the embryo to check where the notochord cells are. This experiment overall helped us validate that our clustering was what we would expect to see based off of known Ciona biology. And it also shows that in situs of known and unknown markers uncovered by our analyses can help us validate our clustering results. While looking for unknown cell types, we noticed that one of our sensory vesicle clusters contained a marker that was highly specific to a region. This marker was annotated as ARX, and it located to row 3 of the neural plate. From this violin plot, we can see that ARX isn't really expressed in any other clusters and only within this particular row of the developing neural plate. When we looked for ARX on Ciona databases at the time, we couldn't find any information, and it seemed like this gene was only annotated as ARX due to sequence homology with other species. So as a first step, we tried to see what ARX was. ARX stands for Aristotelis-related homeobox, and it was first implicated in Drosophila for limb formation. It's expressed in the embryonic forebrain of zebrafish and mouse, and mutations in the human ortholog of ARX are associated with mental retardation and epilepsy. This was interesting to us because the sensory vesicle within Ciona, which is what row 3 of the neural plate develops into, is related to the vertebrate forebrain. It's the anteriormost part of the central nervous system for Ciona. And while it's known that chordate brain development is highly conserved, this finding of ARX hinted at the degree of conservation across these species. So just to make sure that what we were seeing with ARX was real, we wanted to evaluate the localization of ARX to another known marker known as MyTF. MyTF is known to be important in the developing A lineage of the mesenchyme and nerve cord, but also this particular row of the anterior sensory vesicle. So from the single cell map, we can see that the expression patterns of ARX and MyTF align on row 3 of the neural plate, but ARX is very lowly expressed in this region compared to MyTF. Now that we've confirmed that these two markers overlap from the single cell map, we wanted to go back into the lab with the help of Hannah to perform more in situs. So here Hannah's pictures of MyTF and ARX. On this first row, I have the zoom in of the embryo because row 3 is fairly small. And then here I have the two overlays. We can see that ARX and MyTF both line up in the same location, confirming our suspicion that ARX is a highly specific marker for row 3 of the neural plate at this time point. After we found this marker within our cluster, Mike Levine's group at Princeton also released a single cell study within Ciona, and they're able to find the same results. This shows that we have an accurate single cell map that will help us determine the contents of each cell that are important for helping them progress through Ciona development. So in summary, I was able to generate a high resolution computational map of the Ciona late gastrula embryo, and we were able to validate the presence of known and uncharacterized cell type markers with fluorescent in situ hybridization techniques for brachyury, orphan BHLH1, ARX, and MyTF. As some future steps, I want to integrate the single cell RNA-seq data from the other time points that we studied, the early gastrula and early neurula stages, to understand the formation of tissues across these stages, such as with notochord development, for example. And I also want to compare our single cell atlas to other developmental atlases to determine what gene regulatory differences or similarities we might have with other organisms, such as Ciona savini, Felusia, zebrafish, and mouse. So with that, I'd like to thank everybody in my lab, especially Ben for a lot of the wet lab experiments, as well as Hannah for doing the in situs. Thank you, everybody.
All right. Thank you so much, Michelle.